So we've already been introduced to photosynthesis and the basics of what it is. But as a little reminder, let's again review a little more. So photosynthesis. Photosynthesis, we are using light energy to produce chemical energy in the form of an organic molecule. Okay, back to a little basic chemistry. What does it mean to be organic? Well, you have to have carbon, hydrogen, and oxygen. So what we're going to look at today is how that light actually gets used to start this reaction. We actually won't form the final molecule. We won't form our final product, which is our carbohydrate, our CHO, our glucose molecule. But we'll actually see some intermediate steps that will lead us to this reaction, or to this final product at the end. Just like in respiration, all of this isn't happening in one step. Okay, there's lots of steps that happen in between. And a lot of it will actually look very similar to respiration. So we also had talked a little bit about light energy, keeping in mind that only visible light, the smaller portion in the light spectrum, that can actually be used. Visible light, there's a range of short wavelengths to longer wavelengths. The longer the wavelength, actually, the lower the energy that you're receiving from that wavelength. Then we also talked about, well, how do plants absorb that light? Well, in plants, we have our pigments. Chlorophyll A and chlorophyll B, mainly, are the big ones, that are actually used to absorb light. Where are those pigments found? Well, those pigments are found in the chloroplasts of cells that can actually absorb light. So only in cells that receive light. Let's think about what that means. If I have the roots of a plant that are always underground, well, are those roots green? Do they have chlorophyll in them? If they had chlorophyll in them, would there be any use for the chlorophyll? So if you think about cell specialization, is it worth making the chloroplast to have in the cells of something like the roots of a plant that's always hidden underground, if it's not going to be used? Okay? When we're actually looking at the chlorophyll molecule, this should look very familiar to you. It's very, very similar to a lipid, a phospholipid, part of the phospholipid bilayer. Here at top, you have the head of the molecule, and in the head of the molecule, this is actually where you absorb the light. And the tail of the molecule, it's made of hydrocarbons. Well, are hydrocarbons polar or nonpolar? Which one are they? Hydrocarbons, let's remember that they are actually nonpolar, and that's actually what's going to allow them to stay within the membrane, or inside of the membrane, of this thylakoid membrane. Okay? So you have chlorophyll inside of the thylakoid membrane. Well, what are thylakoids? Thylakoids are the actual stacks that are found inside of your chloroplast. A whole stack is called a granum, and all of that is inside the two membranes of the chloroplast. Remember, this structure is very similar to mitochondria. Has its own DNA. We thought that it was a prokaryotic cell at one point, a free-swimming, free-floating bacterium at one point. And then we looked at some of the absorption of light that can be done by various pigments in a plant cell. Here are various pigments, different proteins that can absorb light. Notice that most of them, well, the two that we'll mainly concentrate on is chlorophyll A and chlorophyll B, but very few of them, if any at all, can actually absorb green light. Okay? When we see a color, that color is actually the light spectrum spectrum, the light wave, that's actually being reflected. So for plants, if you only gave them green light, well, they're going to reflect the green light. So that's what we see. When it reflects the green light, that means it can't use that light. That light wave will not be used and will not be able to be used in photosynthesis. So it's not really an efficient light for the plant. But moving on to today, we're really going to focus on the light reactions. And a lot of times you'll hear this called the light-dependent reactions. You need light to power this process. So before we even get started, let's look at this image. What's all here, okay? Well, on the outside, you see this arrow showing you the energy of electrons. What is it indicating? Well, as electrons move up in this diagram, it's high in energy. But as it moves back down, that means it's going to decrease in energy. Question here, what do they convert light into? Well, here you can see light being used in two places in this reaction. What is the light doing? Well, the light is actually going to be used to excite electrons, okay? So let's start at the very beginning. The beginning of this process actually starts at Photosystem 2. Photosystem 2 actually contains chlorophyll A. Kind of weird because we start with Photosystem 2 and we'll end with Photosystem 1. And the only reason why they're labeled like that is because someone discovered this photosystem first. So that's what's stuck, okay? But really what you should pay attention to is the types of chlorophyll. A, okay, that's where we're starting, and B we'll use later on. What else do we see in this photosystem? Well, within the middle of this, you see a P680. P680 is actually referring to the light wave that's being absorbed here. So the light is actually being used to excite electrons that are already in this photosystem. So notice that here we have two electrons being charged, being excited, and as they're being excited, they'll move up to this primary electron acceptor. A little different from an electron carrier, okay? Over here, number 2, well, what do we see happening? It says the splitting of water releases oxygen. So notice here you have water. Here you actually have two electrons that will actually be used to replace the electrons that are excited by the light. So another little cycle within a cycle, okay? Every time we use some electrons from this photosystem 2, now it has some empty space for new electrons to be released. So we'll break down a water molecule, get 2 more electrons, and continue this process over and over again. As we break down water, we also receive some oxygen here. And the oxygen and the 2 hydrogen ions are also produced, okay, from that reaction. But let's focus on that electron, because remember that electrons are what powers the system. So as our electrons move up, they're high in energy. As they're high in energy, they're actually going to move through an electron transport chain. This is going to be very similar to what we saw in respiration. These electrons will move through various proteins, okay? PQs and cytochrome complex, another PC, all electrons creating this gradient. Notice that it does tell you in step 4 that the electron flow provides energy for chemiosmotic synthesis of ATP. We've seen those words before, chemiosmosis, and how we synthesize AP. And a little bit at the bottom, we'll actually go into a little more detail about how that actually happens. But as electrons flow, well, now they're going downhill, and as they're going downhill, they're losing some energy. When they lose energy, they'll actually be received by photosystem 1. Photosystem 1 contains chlorophyll B. And now that they're low in energy, we'll actually take more the light wave P700, and we'll actually excite those electrons again, goes up to another primary acceptor, goes through another electron transport chain, a slightly smaller one, and we'll actually donate those electrons to the carrier, NADP+. And that will form our final electron carrier, NADPH. Okay? We do get one of those hydrogen ions that join with it from that water that we split at the very beginning. This picture right here, a lot of times will actually be referred to as a Z scheme just because of physically how that looks. Okay? And now we want to focus on this electron transport chain. So when we look at the actual electron transport chain, this picture is very, very similar. You'll want to take a minute to compare, pause the video, and compare what we saw in the first figure, first diagram, to this diagram. How is it the same? How is it different? So again, here we're still talking about our light-dependent reactions, and we're focusing in on the chemiosmosis, okay, the electron transport chain. What was chemiosmosis again? Well, chemiosmosis was a way to generate ATP using energy from a proton concentration gradient. Okay, and we did this before in respiration, pumping up those H+ ions, moving them up and down through the system. When we make ATP, this process will be called phosphorylation. We'll actually be using the energy from this process. The NADPH that we're making will be actually making through electron energy. Okay. So here, what do we have? Well, we have our photosystems again. They look a little different in this diagram. They're showing you within the membrane of the chloroplast. We're actually within the thylakoid membrane of the chloroplast. And what do we see happening? Well, here's light charging the photosystem. We know when light charges the photosystem, we're going to excite some electrons. Water is also being broken down as a little side reaction. As we charge those electrons, the electrons will move through the proteins. Here are our different protein complexes, PQ, PC, cytochrome C. And as we actually move through the cytochrome C, notice that here we have our proton. That's our proton gradient, the H+ ion. This proton will actually move on and go to another area where we have another ATP synthase protein. Okay? Very similar to respiration. There we'll actually phosphorylate. Here's our inorganic phosphate and our ADP, and that will be converted through that mechanism, turning of the mechanism, to ATP. And so this is actually called photophosphorylation. Okay? We use light energy to phosphorylate this ADP, giving it that phosphate here to form ATP. But what happened with that electron? Well, that electron continued to photosystem 1. Photosystem 1 charged it up, excited the electron again because of light, went through another electron transport chain, a slightly smaller one, and then we go to another protein where eventually we form our ADPH, okay, our electron carrier. Both of these products, notice they go to the next step, which is the Calvin cycle. So then, what do we have down here? Cyclic phosphorylation, and this is referring to regulation. What I want you to look at here or pay attention to is that this little area right here, the electrons that are received by the photosystem 1 that get pumped up by the light that's charging it, the electrons can actually be cycled. Okay? The electrons can actually be cycled. And so this is the cyclic portion of photophosphorylation. Once it gets to the primary acceptor, it doesn't have to go right away through that second electron transport chain, but instead it can actually go back through the system, back to cytochrome C, back through this other can go ahead and label it ETC2 and ETC1 because that's what we would first see. You can actually go back and use that electron again. So this will happen, this cyclic phosphorylation, photophosphorylation will happen when we have a high need